Today we're going to be looking a little bit more at the brain, but more at the old brain, I suppose, as well as then moving on to the spinal cord as part of the central nervous system, and then finally into ways that we find out information about the brain. Okay, so once again we're back at the old Genes to Cognition online. Which is has a great little 3D brain. Also has an app as well, which you can grab from the iTunes store, or I think it's on Android as well, hopefully. So today we're looking at the brain stem, okay, as well as the thalamus, which you'll see in a minute, which lives up there, okay, all part of what we call the reticular activating system. Your textbook has us on page 203 if you want to follow along. But what we're talking about here is, oops, wrong way. What we're talking about here is the brainstem. Okay, these are the older parts of the brain. Most animals, if not all animals, all mammals anyway, have this part of their brain. Okay, what this part does is this is strongly in control of our alertness. Okay, it's also in control of a lot of the things that we don't actually think about. Last video we talked about in the cerebral cortex was all things that we had conscious control over, things like our movements and all of those kinds of things. This part of the brain is still involved with those things, but it is also very highly involved in keeping the body at a normal state. We call it homeostasis. You can see that here. It says that the functions are maintaining homeostasis, which is keeping things level, keeping our breathing normal, keeping our heart beating at a normal rate, our blood pressure at a good pressure, those kinds of things, as well as our alertness and our balance and our sleep, I suppose, our awareness. So there are three major sections here. You can see the medulla, the pons, and the midbrain. Also, this section here, the cerebellum, is also part of the old brain, the hindbrain. Okay, but we're not too concerned about that at the minute. What you see here is the beginnings of the spinal cord. Okay, the spinal cord would continue down through this section, and all the way down your back. And that's about it. That's the main things we need to know. In your textbook, it talks about the reticular activating system, which is basically our alertness and awareness and our sleep. There have been some very interesting studies done about it where they have stimulated certain parts of this in certain animals, and that has made, for example, they did some stimulation on cats that were asleep. They stimulated that part of the brain. The cats immediately woke up and were very alert for a period of time. By the same token, if they cut this part out of the brain, cats then slipped into a coma for a long period, long enough until they died and they never woke up again. So this is really important to keeping us awake and alert, but then also making sure that we get our sleep and our rest, because we know from our last unit how important sleep is to us. Okay. You can see now a little section in the thalamus. Okay, we'll just zip around here. You can see this little structure in here. All right. And that sits atop of the brain stem. Okay, so you can see there it's at the top. Again, very important for our alertness and our sleep-waking patterns, as well as other things like our memory, consciousness, and perception. Why is this so important? Well, oops, I'll go the other way. Because this is kind of like, it's like the major intersection in the brain. It's the major part where all the information comes in and all the information goes out through this road. So if you have sensory information, so information coming from information coming from your eyes, so you can see me about to draw on this thing. You can also hear my voice. You may be able to, depending on where you're watching it, if you're on the bus or if you're at home, there might be a TV on, or you're sitting on your bed or your couch or your chair. you can feel all those things. All that information comes up through the brainstem into this area, and then it is sent to the appropriate areas of the brain. So if it's sensory, it might require information in the somatosensory cortex. Okay. If it's vision, so if you're watching this, it's going to send it straight down to the occipital lobe. And if you're hearing my voice, then it's going to keep it in here in the temporal lobe. Okay. Once that information is then processed, and if there's any action required, then of course it's going to come from the motor cortex in here. Okay, it's going to go then back out through the thalamus, through the brainstem, and then out to your arms, your legs, and any of those things that require movement. Okay, so this is a very, very important area of the brain because it gets a lot of traffic. Okay, if there's damage to this area of the brain, you can go blind, you can go deaf, you can struggle with some movements. And that makes sense because this is where all of those things come through. It's like creating a roadblock, and of course cars wouldn't be able to get through this way. Sensory information or motor information might not be able to get through. One other thing, as we said before, very important with our alertness and our consciousness. You know, with our consciousness, one of our major altered states of consciousness is sleep. One thing we do know about the thalamus is that it is heavily involved in sleep regulation and our awareness. We know this because of a pretty horrible disorder called fatal familiar insomnia. What does that mean? Well, fatal, of course, death. Familial is family, and insomnia, no sleep, is a genetic disorder that unfortunately some members of the world have. Not a lot, it's pretty rare, but those people that are all related have a thalamus that basically starts to shrink and disappear, it degenerates, and as that happens they lose the ability to go to sleep. So initially it presents as kind of normal insomnia. You can read about this on page 206 of your text if you want. It originally becomes kind of a bit of insomnia and discomfort and trouble staying alert and trouble getting to sleep. It then progresses to a point where they can no longer sleep, and as you know, if you don't sleep, you die. And unfortunately that is one of the problems or one of the issues faced with people that have this genetic disorder, and it is heavily based around the thalamus and how the thalamus starts to degenerate. Okay, so they're the major parts of the brain that we're looking at, but I want to track this down to continue looking at another major part of the central nervous system, and that is the spinal cord. As we track down here, you can see that all of this brain starts to come down into one small section. So just so I can see that. You can see that is like a bit of a stalk of a vegetable, of a broccoli or something. That continues down. It continues down the back of your head, and it becomes your spinal cord. Now, the spinal cord is very important. You can see the small sections of it there and there. And it is really important for a lot of reasons, but one of them is it connects our brain and our major functioning with the peripheral nervous system. Now, I'll show you here the reflex. We're going to talk about that in a sec, but I'm going to just talk a little bit about the spinal cord. So, you can see here in our drawing we have the brain up here and then the spinal cord coming down here. Now, that's usually protected, or always protected, by our spine, our vertebrae. If you kind of touch your neck, usually about your shoulders, you can start to feel your backbones. Okay, that's our spine. That allows our spinal cord to be protected as well as get information in and information out. What does it do? Well, in a nutshell, it's the superhighway to the brain from everywhere else in the body. If you have someone, tickles your neck, okay, they tickle your neck here, sensory neurons take that information to the spinal cord, okay, and they shoot it straight up into your brain, and your brain says, I like this or I don't like this. If that's the case, okay, the motor cortex will then send out another neural message back this way to say, neck muscles move, and you'll shift out of the way, okay? Same goes for everywhere else. Same goes for if you want to lift your arms, okay, then you need information from the brain, the motor cortex, down the spinal cord, out to your arm muscles, okay, to either contract or to extend, and that will move your arms. Okay, so next time you're waving at your friends, that's what's happening there. All right, all very important stuff. We know this because we've seen people that have damage to this area. So for example, if someone is in an unfortunate accident where their spinal cord is cut, severed, we know that that usually turns them into a quadriplegic, meaning that any information that the body gets from below that point, okay, so say information from the shoulder or information from the ribs, or if you're getting tickled by someone there, or the arms, okay, any of that information, once it hits the spinal cord, it gets up here, it cannot get to the brain, which means that it's useless. So that's why people can't feel anything if they've had a major spinal injury above, around the neck area. That's why they can't move and they have those wheelchairs. Same idea if there is some sort of injury, let's say, in the middle of the back or at the lower of the back because of a car accident or something like that, then they would lose all sensation below that area, okay? Meaning that they lose the use of their legs, they couldn't feel their toes and those things. They would still be able to use their arms, most likely, depending on where it was, the damage was, and that's what we call paraplegia, okay? So really important stuff. The other thing is that we need to remember, we need to know that this is part of the central nervous system, or CNS, okay? So the central nervous system is the brain and the spinal cord, okay? Very important. The reason why the spinal cord is so important is because it connects our central nervous system with our peripheral nervous system, our PNS. And you guys have already done some work on that, but there'll be another video of that coming up very shortly. But the reason why, yes, as I said before, the spinal cord is so important is because it connects the PNS with the CNS, all right? Now, what you also have here is a little diagram of a reflex action. Because the brain and the spinal cord are still connected, and the spinal cord has some ability to make very quick decisions. So for example, let's think about this. I'm going to draw a really bad fire. Okay, so you guys all know that I'm a really bad drawer. Okay, but there you go. It's a fire, and I'll put some little logs there. It's like a bad version of paint. So there's the logs, and there's the little fire. So let's think. There's a bet or something, or someone dares you. They say, "We'll see how long you can hold your finger over a flame, over a candle or a fire." What happens is that at some point it gets really hot, and your brain, well, not your brain, if you put it straight into the flame, your hand will automatically pull back before it realizes, ouch, that hurts. Okay, so if you put your hand on a stove, something like that, it's not always hot, it could be cold as well, you will find that your body moves before your brain realizes, ouch. Okay, and that happens for this reason. It's what we call a reflex action. So you can see here that the message from your finger will pass down this line, okay, and up into your spinal cord. Now what happens there is, because there's such an intense pain, your body could not afford to wait the tiny amount of seconds it would take to get to your brain and then back down and say, ouch, that's hot, and then back to your muscles. So what happens is your spinal cord can do a bit of its own thinking. It's not really thinking, but it can react to this situation. So it uses these interneurons here to create a motor message to say, move the arm. We'll process this later, okay, but move the arm. So in essence what happens is the sensory message comes through the sensory neurons to here. The interneurons, okay, transform that message and they turn it into a motor message to say move the arm. At the same time, that message continues up the spinal cord to the brain, where the brain then computes the sensory information to say ouch, okay, and that's where you then will start to feel pain, and you'll also, your brain will start moving into actions like waving a finger around to try and cool it down and those kinds of things, okay? But that's a reflex action. That's where we see the sensory neuron, interneurons make the connection and a motor message. The motor neurons are then acted before the brain is actually put in place, okay? And these happen all over the body in a number of different ways, but this is the most easy one to remember. So if something very painful happens, you touch something really hot, your hand will pull away well before your brain says, ouch, that's hot, okay? So that's the central nervous system, really important part of our functioning, the most important part of our functioning, to be honest. And it connects with the peripheral nervous system, which you're going to hear about in the next video. But before we move on to that, I want to go into some research methods of how we know about the brain. So we have fantastic images like this that come from genes to cognition, but what we have to know is, how do they get these images? How do they know? Well, okay, so brain research methods, page 237, how we study the brain. How do we study the brain? Can't see it unless you chop open someone's head, poke around. That's a bit rude. Also, most early studies of the brain were done on dead people. That's not much good to us because they can't tell us what's going on if we poke stuff and cut stuff out. So for a long time, brain research was hard work. Okay, we didn't have fancy little 3D models like this that we could flick around on the internet. So how did we do it? Well, initially, a lot of the early brain stuff was dealing with volunteers, or sometimes not always volunteers, but people or animals, where we could actually get access to their brain while they were still alive. Sounds a bit gross. Sounds like it's probably in a horror movie. But, in the name of research. So, initially we would have ways of imaging. So we had x-rays and stuff like that, but they don't tell us a lot. We developed the idea of EEGs. Okay, so they're kind of important. But to find out which parts of the brain actually did things, we needed to find specific information about how it all works. Now, we figured out that electrical stimulation or electrical current is an important part of the brain. We know that from EEG, electroencephalograph. It measures electricity. So we know this. So why not put our own electricity into the brain and see what happens? That's what they used to do. A thing called direct brain stimulation or electrode stimulation. What does that mean? Well, what does that mean? People used to be researched on, and what you could do is that you could cut open their head to see the top part of their brain, and you could get a little electrode, like a little pen zapper thing, and you could zap them, I suppose. And if you zap them in certain parts of the brain, different things happened, of course. If you zap them in the occipital lobe, they would see flashing lights, or sometimes they would see colours. If you zapped them here in the temporal lobe, they might hear sounds. Yeah, they might hear a doorbell or hear a siren, something like that. If you zapped them in the somatosensory cortex, in the parietal lobe, if you zap them there, they might have a tingling sensation in their fingers. They might have a tingling sensation in their stomach or wherever it was that you were putting your little electrode zapper. If you zap them in the motor cortex, you might get them, they might move their arm or twiggle their fingers. You know, they might blink their eyes, depending on which part of the motor cortex that you zapped. And this is exactly what they did. Researchers, a guy called Penfield was big in this area, and he would get the tip of an electrode and he would, you know, zap people's heads. It allowed him to create a map. You know, if I zapped, see if you follow in here, if I zapped up here, then maybe someone's finger moved. If I zapped down here, then maybe their elbow twitched. If I zapped all the way over here in the motor cortex, maybe their toes wiggled. Okay, he was able to map certain parts of that. Okay, as well as the same kinds of things here. If he zapped the occipital lobe, sometimes they saw color. Zapped another part, sometimes they saw bright lights, those kinds of things. It was a very, very interesting study, and he did quite a bit of work on this. Okay, but the problem with that is you have to chop someone's head open, and that's not always cool. And then once you do that, you zap them with electricity. So getting volunteers for that can sometimes be a little bit difficult. Could you imagine signing up for that? Excuse me, miss. We're just wondering if you would help us out with some research. Basically what we want to do is we want to chop your head open like half an egg, and then we just want to poke you and give you little electric shocks and see what happens. Not always going to work. But anyway, that was Penfield, and he did some really cool stuff, and we learned a lot from that. Now, so electrode stimulation. Give you this little graph here. This is an advantages and limitations. I'm going to do this for each of the little methods. You can see here the advantage of it's effective. It worked. It was useful and reliable. Okay, you can't cheat that. If someone zaps your brain, you can't fake it. It gave us a great understanding of the brain. Okay, and it improved our research abilities because it started to give us a map. Once we had a map, then we could use some not so intrusive or invasive methods. So it's risky, it's unethical. Sometimes it's hard. Not everybody's brain is exactly the same. It's pretty close, but not exactly the same. So, and usually, you know, not normal functioning brains. What does that mean? Well, try and think of the people that you would be able to use for this study. They might volunteer if perhaps they were really sick, or they were mentally ill, or they were, you know, not in a good way. So sometimes these brains weren't normally functioning brains, because normal functioning people would be like, hell no, you're not sticking electrolysis stuff in my brain. Go away, you freak. So that's that. So what does that mean? Well, it's still a pretty reliable way of doing things. So without chopping people's brains open, what could they do? Well, they could use a kind of magnetized way of doing the same thing. Have you ever used, sometimes in science you do that really cool trick where you get like lead shavings and you put it on a piece of paper and you get a little magnet and you run it around under the paper and the magnet's shavings follow it? Have you ever seen that stuff? Hopefully you're nodding and you know what I'm talking about. The same kind of idea came through some other work done called transcranial magnetic stimulation. Basically, your skull is good from protecting you from small falls, but it is pretty good for allowing magnets to move information through it, just like, you know, like a piece of paper. So, what do they do? Well, it's very similar to the electrolysis stimulation that we just talked about. So what happens is they use a magnet, okay, to kind of shoot through an electric impulse. So they get a little coil and they might sit it, say, over the occipital lobe here, and then they put a little charge through the coil and it enters a magnetic field created by the magnets, and they shoot that magnetic field through the skin and through the skull and into the neurons below. Sounds a bit full on, but it has a similar effect to electrically stimulating the brain directly. So the same idea, flashes of light, the same kinds of ideas were found. It was very similar, except that you didn't have to cut people's heads open. So it has its benefits as well. If we go to our benefits and advantages is here, it's non-invasive, can't be used if there's metal or medical devices in the body because they'll get attracted to the magnets and they might shoot out of you. We'll talk about that when we get to MRIs. Again, there's no hole in your skull, which is good. You don't need any anesthetic. It's more ethical because people could say, yeah, I'll be in that because you don't have to chop my head open. The other thing too is that one of the limitations is it only gets the parts of the brain pretty close to the skull. You can't get too deep in with the stimulation of the brain there. So it kind of just works around the top parts of the brain. And as we know, the brain is a 3D object, and so there might be some other more interesting parts below the surface that that can tell us a lot of things there. So that's the electrical stimulation, also transcranial magnetic stimulation. These are ways that we can stimulate the brain to find out which parts are kind of in control of which things. Okay, that was early studies. They were early ways of thinking about how to get in touch with dealing with the brain and finding out exactly what it is that the brain does. And of course, they use that in conjunction with EEG brain research, which you know all about, the you know, how we found out about the alpha waves, the beta waves, those kinds of things. So what happens then is there's evolution, okay? There's an evolution of technology, okay? Because what we're looking for is this thing here, if you can see that, non-invasive. We want to learn as much as we can about the brain without chopping people's heads open or chopping animals' heads open and going in there and digging bits out and zapping them. We don't want to do that. So what we have is the idea of a CT scan. Now you would have, if you watch House or any of those kinds of shows, sometimes they use the CT scans or the CAT scans because it stands for computerized tomography or computerized axial tomography. Okay, those words are up there, you can see them. Fancy words for computerized x-rays of the brain. And that's basically what they are. They take little slices. So you can see here, this is a cross-section of the brain looking from the top down. So someone's eyes, most likely, yeah, their eyes would be here, okay? And what they've done is that they just take little slices here and there. So basically, just to give you I'll get rid of that. Boom. So if you were to look at this like side on, I suppose, again, really bad drawing, you would have someone's head that looks like this. Okay, it's their nose, their mouth. Oops, I didn't give them a chin. That's their head, right? And their eyes. What this is it takes imaginary slices through the brain, okay? And as it slices through the brain, it takes a photo, and you can see that there. So that kind of photo there would be if we could chop someone's head off and look at their brain like that, then it would be chopped off from there. But we don't have to because we can use X-ray technology to be able to find that out. Now this was really important early on. X-ray was a big deal in being able to see inside the body. But as you can see from these photos, it doesn't tell us a great deal. It gives us some idea of brain structure, and that helps a lot. You know, and that's how we get some images from the fancy 3D brain that we've been looking at. But what happens is it doesn't give us a great picture. It doesn't give us any information about what's happening in the brain. It's basically just like looking at the bones and looking at the arm. It doesn't tell us how the arm moves or anything like that. It just tells us the parts of the arm that are in there. Does that make sense? And so that's that's your CAT scan. And it was very important early on. I don't know if you've seen these, but to go into one of these, and all the other ones I'm going to tell you about with the PET scan and the MRI scans, you kind of go into a giant tube. You have seen it on TV. It's a giant tube, and you're kind of on a bed, and the bed kind of slides into the giant tube. And so with a CAT scan, X-rays, so the X-ray machine moves around you. That's the CAT scan. It's safe. You know, you can identify precise locations and damage, because it's like an X-ray. You can see damage. You can observe changes and differences. So if you take a CAT scan when someone's 20 and a CAT scan when someone's 30, if there's been some sort of damage or degenerative disease, you can see that. The same way you can with an X-ray. It's like a really cool X-ray of the brain, the CAT scan. And at the time, it was very, very important. But we moved on. So we moved on to PET scans. Now, PET scans are pretty cool. Check these out. So what do? Well, it's a fancy name for, well, PET is an acronym for positron emission tomography. You can see it there, positron emission tomography. And what it is that it helps us to reveal where activity is, almost in like a 3D type arrangement. So this idea came along in the late 70s, and what it did was it gave us images of the working brain. So it used a dye, a radioactive tracer. So that's like it's kind of it reacts with things. It's not necessarily 100% safe, just quietly, but we'll talk about that a little bit later. But they inject a radioactive dye into your body, which then moves through and it connects itself to certain parts of your bloodstream. So it flows through your body like your blood. The idea was that just like exercise, the parts of the brain that were working hardest would need the most blood pumped to it in order to get it moving. The same way that, if you go to gym and do lots of push-ups, then your chest gets sore, your arms get sore, because all the blood is pumped into that area. You can see here, I don't need to highlight it too much. You can see here, hearing words, left side of the brain, really the red section is highly, highly active, and the yellow also very active, blue bits not so much. But you can see here around Wernicke's area, remember Wernicke's area, where we're comprehending speech, highly, highly active, lots of blood flow there. When we're speaking words, okay, we can see here in the motor cortex, in the frontal lobe, what area is that? Broca's area, okay, because we're producing speech, so really red bits there. Frontal lobe, okay, we're making lots of associations here when we're thinking about words and seeing the words, of course, in the occipital lobe at the back. So the advantage of this is you get to see which parts of the brain are working really hard, okay? And this gave us a major insight into people's brains and what they were doing, okay? And so what are the positives and the negatives of this? Well, you get detailed images of the brain and its functions, okay? So you can see which parts of those are actually working nice and hard, okay? If I just put that on the side as well. So we can see that at the same time, right? So we can see that the left side of the brain is working really, really hard when we hear words. It's easy to determine active areas because, we've assigned colors to high density blood is red, low density blood is blue, so on. And also, it took the focus away from brain damage to brain function. That's really important because for a long time, brain research was done with damaged brains. Okay, and now we're moving into functional brains, healthy brains. What does a healthy brain do when we hear words? Well, it does this. Okay, that's important. Also, what are the problems? Well, we don't know if it's caused by mental process or behavior. Okay, you can only study short tasks because this stuff in here is a radioactive dye. That's not the blood, it's the dye connected to the blood. If you get people to think about words or hear words for 5 hours at a time, that means there's radioactive dye in that same part of the brain for a long time. I don't know about you, I don't want radioactive dye in the same part of my brain for 5 hours because, as it says here, we can have some decay. Also, you can't do regular studies all the time because, again, radioactivity. This stuff that you're putting into your body, it's not supposed to be there and can cause some real problems later on. So that's the PET scan, and pretty important and impressive stuff. You also have another option called a Spect, or single photon emission computer tomography. This is similar to the PET scan, but they found a way to use a less radioactive dye, which allowed you to do activities over a longer period of time. That's basically all you need to know from that. So the radioactivity level is less, you can do things over more time, but the problem is because it's not so radioactive, you don't get as clear a reading. And if you don't get as clear a reading, you can't necessarily tell exactly what's going on. So it's not as good. And that's the Spect, S-P-E-C-T, which stands for, as I said before, single photon emission computer tomography. Big fancy names. You just need to know Spect. All right, the last two, the MRI and the fMRI. Okay, the MRI stands for magnetic resonance imaging. This is a great little word. And the magnetic resonance imaging is basically like, this is weird, so get your head around this. It's like using magnets to create small vibrations of the atoms in your brain, and then it reads the vibration of those atoms to create an image. Kind of like that. Not kind of like that at all. But that's the same kind of idea. It looks a bit like an X-ray in some regards, but it's a lot more colourful. It's basically a more clearer picture of than we had with the CAT scan or the CAT scan, and it gives us like a really good picture of the brain. But the MRI doesn't give us, like the CAT scan, doesn't give us anything about function, okay? But they delivered on that a few years later with the fMRI, which you can see here. So what you have underneath here is an MRI photo, which looks, as you probably figured out, looks a lot like a CAT scan, but it is in more detail, and we can move through the brain a lot better. It's a clearer picture than the CAT scan, and you know, new images now are, you know, it's like 3D. Basically, you can get images of the brain, fMRIs, look like this now. Okay, so this kind of understanding of the brain comes from MRIs and fMRIs. Okay, so finally, the functional magnetic resonance imaging tracks the oxygen levels in the brain. Now, what does that mean? Well, of course, you have your blood carries oxygen around. That's what it does. Okay, so the more blood in a specific area, of course, there's going to be more oxygen in that area. Okay, so what it does, a bit like what the PET scan did with radioactive, where they put the radioactive dye in, okay, the same kind of thing applies here. So the good thing is there's no crazy radioactive goo in your body, okay? They can just detect the oxygenation levels of the blood and therefore what's going on. You can see here the same idea as the PET scan, that really red levels are highly active. In this case, the really yellow bits are highly active. The blue and the green are not so active, and if there's nothing, then there's no activity. Okay, so obviously something's happening in someone's frontal lobe here on both sides. So you know, that could be anything. That could be an emotion, could be something like that. But the fMRIs are probably the newest technology that takes on board all the others. Okay, so you can see here, this looks like the early CAT scan. Okay, but it's clearer. Okay, it's non-invasive. It looks like the PET scan, okay, where it measures the activities in the brain, but you don't have to have a radioactive goo in your body to make that happen. Okay, so the fMRI is now usually the most widely used if you're going to track brain activity, and here are the reasons why. It can detect changes moment to moment as they occur. So you put someone in the tube and you might play them music, or you might give them a maths problem, and then as they think, as the blood moves around their brain, then you can see where it moves in real time. Okay, it's very detailed and these days very precise, and they can create fantastic maps of the brain for thinking and those kinds of things, right down even to individual blood flow patterns. So what are the limitations of it? Well, in a normal functioning brain, you cannot assume cause and effect relationships between activity and mental process. Okay, just because one part's really active doesn't necessarily mean that it's solely responsible for that. Or we don't really know if it's the major response. It might just be a lot of activity goes to that area. So still not 100% sure. Also, we cannot know if activity is caused by the actual task or by other factors. So if one part of your brain lights up, we don't know if that's responsible for your hearing, or that's responsible for hearing things that are only 30 seconds long. What if if you have to hear things over an hour? Not sure. But there's slight limitations. The fMRI is very important in the way of brain research. So, what have we talked about today? Well, we've talked about the brainstem and the thalamus, which are really important to regulation of the body. We've talked about the strengths and limitations of ways that we study the brain, which are really, really important, and you're going to need to know both of those. So make sure you look at the strengths or the advantages and the limitations, okay? And you will know as we went through them, we progressed to less invasive, so less kind of in your brain, and more clarity around what we actually understand about the brain. All right. Next video is going to look at the peripheral nervous system, because now we've covered the central nervous system. So I look forward to teaching you guys about that. All right, that's it from me. I'll see you guys later. Bye.